Hi, I'm Olivia Rader, and I'm here to teach you about neural transmission. Now to get started, for the first slide we have functions. As you can see, I have a little image on the side that kind of shortens everything up if you want to take notes on that. But as shown in the picture to the side, the dendrites receive information from other cells, and the cell bodies are what keeps everything organized and keeps the neuron working correctly. The axon hill creates the impulse. The nod and Ranvier gives the okay for the diffusion of the ions. So when the gates open, which you'll learn here shortly, that's what it creates, or it okays. And then you have the squalene cells produce the Lehman sheets, which helps move the signals faster. The nucleus is what controls the neuron. The axon transfers the signals to the other cells, and lastly, the axon terminal is what helps the junction from the nearby cells. All these parts are needed for the neuron to function correctly. Next, you have the electron portion of the action portion. You have the major ions involved are the sodium, which if you see in the picture, it's going to be the Na with the plus sign, and the potassium, which is shown as the K plus. The rest of the membrane is where the neurons have more positive charge ions outside of the cell rather than the inside. To have the resting membrane, we will need more sodium ions outside than inside, and more potassium ions inside than outside. And I know it's kind of mouth wordy, so if you look at the picture onto the side, it kind of shows you and demonstrates what that means. Next, we have the changing of resting membrane, and as you can see, you'll find the rapid changing in the resting membrane potential that creates the action potential. This is created by the potassium gates being open, which allows potassium to rush in uncontrollably, which then can change the membrane potential from negative to positive. And as you can see in A, B, and C of the image, it kind of demonstrates each phase, I guess you could say. So as you can see, the negative 70 and Na right there. All right. Next, we have chemical portion of the action potential. After releasing the synapse cleft, you start to see neurotransmitter receptors binding the receptors, which is also called the neuroreceptor. This is where the neurotransmitter mixed with the proteins on the membrane of the synaptic cell. This then causes the channels to open and close. When the channels open, the depolarization happens when creates another action potential and changes the membrane potential. So as the gates open and close, you can see in the photo the molecules moving in and out. This is what makes it either a positive or negative, which if you go back to the back slide, you can see that's what creates the resting membrane. And we'll go to the next slide. Release and removal of the neurotransmitters. So as you can see, I also have the image off to the side showing you the exososis, which we'll learn here soon, and then the endososis, which they're both opposite. One releases and one is what brings them in. So the neurotransmitters are released from the presynaptic neurons from exosis. The molecules are released from vessels that connect with the plasma membrane. As you can see in the image, exosis is the opposite of endocytosis. When neurotransmitters are being removed, they can be removed by being in the synaptic cleft and being destroyed by the enzymes, or it can be recycled back into the presynaptic axon terminal. So they actually will be recycled and reused in the presynaptic terminal. And there is my sources. And that's all. Thank you.